Yes, ma'am.
Okay, so let's start the class for today. So in the last class, what we have talked about is we were discussing about the development of the female gametophytes, right? So আমি কি পঢ়ি আছিলো megaporogenesis or development of the female gametophytes, right? Our megaporogenesis of what we remember that the megaspore mother cell undergoes the meiotic division or episode. It ultimately forms 4 megaspores, and out of the 4 megaspores, 3 of them degenerate and only one remains functional, right? Are functional asile? I think we have discussed that also. The megaspore that is towards the cellagel end becomes functional, right? Are he functional megaspore it undergoes division and redivision, and ultimately megaspore ultimately we have a 7-cell structure, right? 8 nuclear or 7-cell structure. Last class right? We have talked about the female gametophyte also. What is the other name of the female gametophyte? It is nothing but the embryo sac. How does the embryo sac develop? Can it divide nuclei first? Eta nuclei get divided into 2, and then 2 nuclei get divided into 4. Pre-nuclear division eta hoisile, and ultimately after key form korisile, cell form korisile, no? And in the last class, the embryo sac in the structure, we remember we have seen something like this, where we had the central cell, no? Central cell or duta nuclei dekhechilo. Tar pisot if suppose if this is the antipodal cell, we have seen this one as the egg apparatus, right? So up till here we have done. So we are going to talk about the embryo sac today. Female gametophyte or embryo sac. One information the first point is there was a person called P. Maheshwari who has classified embryo sac embryo sac or types disile, right? And what he said, the embryo sac is of 3 types, right? Are on the basis of key. Let's write it. On the basis of number of megaspores participating in embryo formation. So on that basis, what he said is, he said there are 3 types of embryo sac. Or 3 types like here, monosporic, bisporic embryo sac, aru tetrasporic embryo sac, right? So what we are saying is, we are talking about the embryo sac. Embryo sac is also called as what? The female gametophyte. According to Maheshwari, what he said, the embryo sac formation megapore, and I think we remember it from last class. 1 MMC will always produce how many megaspores? It is 4 megaspores. And what we have learned is, out of the 4 megaspores, 3 degenerate kore aru eta functional hoy thake. Now from that point, what we need to remember is, if a koru 3 degenerate kore aru eta functional hoy thake, not necessary it is a statement that goes for each and every angiospermic plant, right? So depending upon plant to plant, no, kibanta megaspore functional hobo, it will vary. But the most common cases, in most of the common cases, what we have seen is, we have seen that the 3 of the megaspores, out of the 4, 3 degenerates and only 1 remain functional. So what did Maheshwari do? He said that depending on how many megaspores are involved for embryo formation, megaspore functional hoy thake, on that basis embryo sac was divided into 3 types. The first is called as monosporic, bisporic and tetrasporic. Monosporic, what we know or what we understand is, only one megaspore formation. I think we understand the word called as mono, right? And examples would be Polygonum, Onothera, etc. Next, bisporic embryos, right? As the name suggests, two megaspores participate in embryo formation. And this is something that is seen in certain plants like allium. Finally, we have the tetrasporic, where all the four megaspores participate in embryo formation. And examples of tetrasporic could be seen in Edoxa, Hamburgo, Fritillaria, Drusa, etc. Right? So this was information according to Maheshwari. Now, out of this, Ami kuntu kori asilu. In the last class, we have talked something about the monosporic embryo development. Now, out of the three types, in detail, we have to talk about only monosporic, because this is the portion that is given in your NCERT book, at least for your higher secondary level. Now, for our further study, we have to talk about these in detail. But as of now, we'll be talking about only the monosporic embryo. So what we have learned is, on the basis of the number of megaspores that take part in embryo formation, the embryo sac could be of different types: monosporic, bisporic, or it could be tetrasporic. We have to talk in details about monosporic embryo sac development. Monosporic Polygonum type of embryo sac. So this was the topic, right? Now we have an idea what is the term, or what we understand by the term monosporic, right? And polygonum is obviously an example of a plant in which this type of embryo sac is seen. Now, what is the reason that we are studying in details only this type, right? Megasporic discuss we generally say that three of them degenerate and one remains functional. That condition is seen in case of polygonum, and polygonum is an example of an angiosperm. And this type of an embryo sac, polygonum type of embryo sac, it is something that is very common in case of angiosperm. And it is into basic common hoy. So that is the reason this type of embryo sac, a polygonum type of embryo sac, is found in almost around 80% of the angiosperm. Next is this development, or this type of embryo sac was studied in monosporic embryo sac to put studied in the plant Polygonum by a scientist called as Strasburger. In Polygonum by Strasburger. What happens here? The nucleus of the flagell megaspore undergoes three mitotic division to produce a 7-celled and 8 nucleate structure. Right? So 3 mitotic division, this is something that we have studied in the last class. Ultimately, what is the product that we have? We have an embryo sac, it's 7 cells and 8 nucleus. So this is the reason why we are talking about a polygonum type. Polygonum type is something that is common type seen in case of angiosperm, whose scientists call it Strasburger. And how does the embryo sac look like? 7 cells and an 8 nucleus structure. Now we have to see what are the 7 cells that we are talking about. The 7 cells are 7 cells asile no. So hatta cell ki asile. The first one, single egg, or it is also called as oosphere. Now we have a term that is called as oosphere. No, I mean that term. No, one is called as a oospore and the other is called as a oosphere. Now don't get confused between the word oospore and oosphere. Oospore is the other name for a zygote. Zygote or coming oospore only called. In the oosphere, we are talking about the egg. Right? So out of the 7 cells in that embryo sac structure, no, it is structure. Suppose we have this one as egg cell. This is as antipodal cells, and this one and this one is the central cell, right? It's our embryo sac. In this egg apparatus, this one is the synergid, this one is the egg, and this one is also the synergid. Right? So so we have the first one, that is the single egg or the oosphere. It has point and a cellagil nucleus. So what we're doing here is we are discussing about the egg cell, no? So if egg is a cell, so obviously that vacuole nucleus, right? So we have a cell that is called the egg cell. Egg cell of vacuole, and we are talking about the cellagil nucleus. Yeah, key question hai, the nucleus position to pin. The nucleus always lies towards the cellagil portion and the vacuole towards the micropylar region. The second point is, it is larger than the synergid. Third is, it shares a common wall with the synergid. But after maturation, or you can say after fertilization, it develops its own wall and forms the zygote. Right? So embryos record, the first cell we are discussing is the egg cell. Egg cell or ki mone rakhibo that it has a micropylar vacuole and a collagenous nucleus. Right? Next is ultimately after fertilization, we know if you're talking from the reproduction point of view, what is important is obviously the male gamete and the female gamete. Male gamete in the pollen grain and female gamete will find it in the embryo sac or the female gametophyte. So ultimately what happens is both of them will fuse and that process is called as fertilization. So after fertilization, what is the product that is formed? We know N into N, no? দুটা এটা মেল গ্যামেট এটা ফিমেল গ্যামেট দুইটা যদি ফিউজ কৰি পেলাই কি ফর্ম কৰে it forms a zygote, right? So the egg cell after fertilization it forms a zygote. A zygote form কৰিব লাগিলে তাত কি থাকিব egg cell এ নিজৰ এখন wall develop কৰিব লাগিব. Before fertilization কিন্তু এগৰ specific নিজৰ wall নাথাকে. It shares a common wall of the synergies. So here I can specific wall nine. So these are the informations about the single cell that is oosphere. So out of the 7 cells, this is the first cell. Next we'll talk about synergies. They are also called as helping cells or cooperative cells. Right? Synergies or information has a micropylar nucleus and a collagenous vacuole. Now you have to pay interest in this line. All of our we have discussed about the egg cell. The egg cell also has a vacuole and a nucleus. The synergid cell also has a vacuole and the nucleus. But what is the difference? The difference is its position. Right? In the egg cell, our nucleus to code or in the synergid, our nucleus to code. In the synergid, the nucleus is at the micropylar end, but in the egg cell, the nucleus is always at the collagenous end. It is a point I'm comparing. Next. Electron microscopic studies has revealed that these cells do not have a cell wall at the collagenous site during maturity. And this is a study that we have seen when electron microscopic studies has been conducted, right? Next is now we have to talk about the important functions of the synergid. No, synergid or function key. Second, this was the synergid, this was the egg, and this is another synergid. So next point is characterized by these cells are characterized by the presence of filiform apparatus. So it was question I have a filiform apparatus. We will see or it is present in the synergid. Now, what is the filiform apparatus or what is filiform apparatus? These are finger-like projections with a core of microfibrils surrounded by a sheath. Right? So, synergies will occur. Whenever we talk about synergies, for example, if we talk about egg, what we remember that the main function is obviously to form the zygote, right? Fertilization of the cell. Next, synergy out of all the points that we'll write or we have written already, what we remember or what we should remember is the main function of a synergy. Synergy, the presence of filiform apparatus is something, or it's a very unique point, right? So, filiform apparatus, right? Finger-like projections. So finger-like projections are then it has a core of microfibrils. Some words called as microtubules, microfibrils, by intermediate fibers. We'll be discussing when we will be talking about the exoskeleton, or we are talking about that part in the chapter cell. Microfibers, microfibrils, intermediate fibers, we'll be talking in details. As of now, we have a word called the microfibrils. Microfibrils is thread-like structure. Hey, thread-like structure at a bunch type. Ultimately, key form for finger-like projection form. Now, what is the function? It helps in absorption and transportation of nutrients. And the most important function is it directs the growth of pollen tube into the embryo sac. So out of all the functions, so this is the most important function. So what we are discussing is we are talking about the synergid cells. No? How many numbers of synergid cells are present? There are two synergid cells that encloses or that surrounds the egg cell. Right? Side synergid segment. Synergid cell, if suppose we are talking about this egg, no? And this one as synergy, no? So a synergy cell finger-like projections develop, and these are called as what? Filiform apparatus, right? Synergic cells are characterized by the presence of filiform apparatus. Now, what is the function of the filiform apparatus? The most important function of the filiform apparatus is to direct the pollen tube, right? Pollen tube, embryo sac of so who will direct? It is always the synergid. In short, we can say that the synergid acts like a traffic police. Means what? I mean, keep right? Suppose this is the embryo sac. The egg apparatus, antipodal cells, central nucleus, right? This is the stigma, and in the stigma, what we know is the pollen grain falls. Pollen grain falls. I think you'll remember that shedding of the 2-cell stage and the shedding of the 3-cell stage. What is most common in angiosperms is the pollen grains are generally shed in a 2-celled condition. And what are the 2 cells? The first cell was the generative cell, and the other cell was the tube cell. No, generative cell or tube cell. Cell tube cell, generative cell. A generative cell will divide and form 2 male gametes, and the tube cell will ultimately form the key. It ultimately forms the pollen tube. Right? So a pollen tube to form, we have talked about it in details. No, this is the male gamete, this is the male gamete, this is a degenerating tube nucleus. Again, we discuss also. And this is obviously the pollen tube, right? So this is something that we see in case of after pollination. pollen tube develop করে. Now, in this side, this portion is called as style. Right? এই style-ডালৰ ভিতৰেদি pollen tube-ডাল সোমাই আহিব লাগিব, and this pollen tube should come and ultimately it should enter the embryo sac. Now, how will the pollen tube know where is the embryo sac exactly located? কোনখিনি জেগাত embryo sac-টো আছে, কেনেকে যাব, right? So, ইয়াতে কি হ'ব, the growth of the pollen sac into the embryo sac, egg-টো reach কৰিব লাগিব, no? সেইটোত কোনে help কৰিব? It is always the filiform apparatus that will direct the growth of the pollen tube. And ultimately, pollen tube main aim ki, it has to reach the egg. So then the male gamete and the female gamete fuse. Who helps in this process? It is the filiform apparatus that is present in the synergy. So maximum question have been asked about the function of the filiform apparatus. Duta question, filiform apparatus is a characteristic of which cell? Second question, what is the most important function of the filiform apparatus? It is the characteristic of the synergy, and the most important function is direct the growth of the pollen tube. And how many synergies do we see? We see 2 synergies. What happens to the synergies before and after fertilization? This is something that we'll discuss in later class, right? So out of the 7 cells, egg was the first cell, and the 2 synergies, ultimately we have discussed the 3 cells, right? Next, we have antipodal cells. Antipodal cells are also called as vegetative cells. Right? They are how many in number? 3 in number. They degenerate before or after fertilization. And what is the function? Absorption of nutrients, right? Nothing in details. Antipodal cells for details, just remember that they are also called as vegetative cells. So what happens to these cells? Gradually, these cells will degenerate. Because we know in the entire embryo sac, what is most important is obviously the zygote, no? So, or in other words, we can say the most important cell is obviously the egg cell. So, zygote to form koribo. And after that, we can also give the importance to that central cell also. Because if you remember, central cell will ultimately go and form the endosperm. Right? I mean, we should discuss it. So, as far as the antipodal cells are concerned, question base antipodal cells are called as vegetative cells. A portion to so 3 antipodal cells, 3 cells in the egg apparatus that make 6 cells, and we are left with the last cell. So, 7 cell structure, our last cell that we will be discussing is the central cell. Central cell kono a gute diagram to a gute portion kiri ami ki boli ko central cell. And if you look at the diagram, which cell is supposed to be the biggest cell in the embryo sac? It is always the central cell that is considered to be the biggest. So central cellular information key is now biggest cell, or instead of using the word biggest, we can use the word largest cell. In the embryo, right? Next is, initially, it has 2 polar nuclei. Polar nuclei, we have discussed it in the last class, which fuse, which fuse together before fertilization to form the secondary nucleus, right? So central cell also plays a very important role, right? The central cell or duta, if you notice this cell, no, it is a cytoplasmic layer. That dotted portion that we have drawn is the amount of cytoplasm that we are discussing. It is not something that denotes a cell. Gote portion to a cell, a cell to ki ase, se duta nuclei ase. A duta nuclei kobra ahile. I think we remember from the development of the female gametophyte, no, megaspore or the functional megaspore cell had one nuclei. That one nuclei underwent what? Pre-nuclear division, no? Pre-nuclear division means if nuclear division is not followed by cell wall formation immediately, that is called as pre-nuclear division. I think you remember this diagram, no? I mean, this was the functional megaspore. And what next? The diagram next was something like this. Eta nuclei per duta nuclei full. And then what we have done is 2 nuclei here, then here. The next step was 1, 2, 3, 4 here. 1, 2, 3, 4 here. In the last class, we have talked about, right? So, what is the next step? We have talked about a structure like this. Out of this 4, 1 nuclei will move to the center. Even from here, 3 will remain at the pole and 1 nuclei will move to the center. These 2 nuclei, these are called as what? These are called as polar nuclei. So initially, a good central cell to polar nuclei thakibo, right? Why it is called as polar? Because this 2 nuclei has descended from the 2 poles of the embryo sac or this structure, right? So hey, duta nuclei thakibo. Kintu duta nuclei ki hobo? They will fuse together to form 1 nucleus. When will such a condition occur? Before fertilization. So ultimately, before fertilization, ki hobo? A because the polar nuclei asile, no? A structure to pisot ki hobo? They will fuse together, and this would be called as secondary nucleus. And one important point about the secondary nucleus is we have talked about its ploidy. What is the ploidy? It is always twice n, no? So central cell to ploidy kiman boli kudile ki hobo twice n. Ki hobo twice n ka duta nuclei fuse korise. Duta nuclei jodi fuse korise, 2 sets of chromosomes, and so we are talking about it as a diploid cell. But if you are comparing the rest of the cell, for example, egg cell or ploidy kiman, obviously n. Synergies of ploidy kiman, it is n. And similarly, if you are talking about antipodal cells bilakoro ploidy kiman hobo, they are always n, right? It is only the central cell that has that secondary nucleus. A secondary nucleus of ploidy kiman, it is always twice n, right? So ultimately, we have discussed the 7 cells of that embryo sac. 7 cells are 8 nucleate because egg apparatus 3 cells, 3 nucleus. Antipodal cells, if you're talking about 3 cells with 3 nuclei, that gives 6 cells with 6 nuclei. The last cell is the central cell with 2 nuclei, right? And that is how it makes the 8 nucleate structure in a 7 cell embryo sac, right? So this is all about the female gametophyte or the embryo sac. I think it's understood, right? Next, after discussing this portion, we need to know certain points from examination point of view. Eta points regarding synergy. Synergy absent. Sometimes, in some cases, the synergy remains absent in plumbage absent. The number of synergies is 1 in number. I mean, generally, in the polygonum type, synergy no? Plant or synergy for example, in peperonia. The next point is about antipodal cells. Antipodal, similarly, antipodal cells absent hobo pare. Absent in case of the embryo sac of Onothera, And number of antipodal cells kiman asile? The number of antipodal cells on that structure that we have discussed was always 3. But sometimes what happens is their number can vary to 20 also. The example being, yeah, maize. Right? So in case of maize, it is 20. So these are certain exceptions that you have to know about the synergic and the antipodal cells. Up till here, what we have discussed in this entire chapter is, since we were talking about reproduction, we have talked about the male reproductive part and the female reproductive part. Development of, we have used certain terms or discussed certain terms called as microsporogenesis and the development of the male gametophyte. Similarly, we have talked about megasporogenesis and the development of the female gametophyte. Once they are ready, I think in the last class what we said is the next step would obviously be pollination. Our pollination or piece of key hobo fertilization. But before going to pollination part, let's see what are the type of questions that are asked on this part. Ketaman questions for him just to have an idea. The A2 portion of run kenuka dhoroner question ahise aru tar pisot we'll be moving to the pollination topic. And we know this chapter is a long chapter, right? So instead of doing question answers at the last, I mean, part of common question answers discuss kori jam. Let's go for the question answers. Okay, the first question is a typical angioformic anther is bilobed, unilobed. It is always bilobed. No? If you remember, bilobe can make something like this, right? Bilobe. And tetragonal consisting of options here are 2 microsporangia, 4 microsporangia, 3 microsporangia, or only 1 microsporangia. So the question has been asked about this anther. No, in case of our typical anther, the anther is always bilobed. Our bilobe structure inside the anther, I mean, keep up. Obviously, we'll get the pollen grains. Our pollen grains, that is what is called as microsporangia. So the question has been asked about microsporangia. What is the question? The eta anther, kuntu anther, bilobed anther, microsporangia kiman thakibo. So ultimately, what is the answer? We have to answer to co-pariban. The answer is 4 microsporangia. So this was the first question. Let's go to the second question. Simple questions is it? Can you confuse hoba? What is the ploidy of the cells of microspore tetrad? Ploidy kiman? So we are talking about microspore tetrads. Microspore obviously any hobo no. So what is the ploidy? It is always the answer is haploid. Options तक लिखा है. Answer is haploid. Let's go to the third question. Pollen grain apparatus called germ pores are present in enzyme intime vegetative cell generative cell. Right? So we have to choose the answer from these options that are given. So we know whenever we talk about pollen grain, obviously the pollen grain has a structure called as a germ pore or germinal furrow. We have already talked about it. It is a question to case that germ pores will have code papa. We know a pollen grain has two walls, the exine and the intine. So germ pore to code as it. Or in other way around, the question could be also like this. Is the exine continuous or the intine continuous? Obviously, the intine is continuous. Or exine continuous kyo no hoy? Kanta germ pore ase. So this answer would be exine. Germ pores bilak we code pam exine or pam. Clear question hai? Question bilak?
Ma'am.
Yeah. Kiba doubt ase nige? Okay. We go to the fourth question. A weed that has become a major cause of pollen allergy. What is the answer? Name a weed that has its greatest contribution in pollen allergy. Pollen for her, we have talked about pollen allergy. There are certain plants that pollen allergy cause for, and what were some of the examples? Parthenium, Chenopodium, Amaranthus, etc. So this question has been asked about Parthenium. Sama option of key option of Parthenium, so we'll be talking about Parthenium. Right? It could be Oenothera also, it could be Amaranthus also. Chenopodium mo hobo pare. Next, a question about a translator apparatus. So question is the translator apparatus code pump. It is seen in the example that you need to remember. Remember the translator apparatus that we have discussed. Something like this we have talked about. No? Remember the diagram? Yes. So this was the translator apparatus that we have talked about. Right? This is something that is found in Calotropis. So examples are also important. Selenium. Next. Intine is made of. Do you have a question? First, exine is made of or intine is made of? Pollen grain, the question has been asked from the pollen grain the part. Exine is made up of key, the most important component of the exine. Poropollenin. Yeah. So, but the question has been asked about intine. Intine is made up of pectocellulose. So, if like question, right. Next, we have certain questions. Questions discuss karu aaj. Okay. The next question is about the function of the pollen tube. Question number 6, okay?
6.
Okay. The function of the pollen tube. Options here. Options. Okay. The function of the pollen tube is to carry. Kiba carry kore no? Ki carry kore? Flagellate sperm male and female gametes non-flagellate male gametes ne flagellate male gametes. Okay. Can you see the board properly?
Yes, ma'am. Yes, ma'am.
Okay. Just look at the question and try to give the answer. Kihor kor question kulise pollen tube no? I think we meant the pollen tube buli kole tat ki thakibo. Male gametes ki thakibo aru degenerative nucleus asile. So what is the main function of the pollen tube? Obviously to carry the male gametes. Etu to ami janu, right? The first option is wrong. The second option is also wrong because female gametes obviously carry no it. We are left out with C and D. The purpose of asking this question is just to know whether the male gametes are flagellate, non-flagellate, motile, non-motile. In case of higher organisms, male gametes bilak ki hoy motile ne non-motile. Tame answer to ki hobo pare? C ba D e bhitor hobo. Male gametes thake. The point is male gametes bilak motile ne non-motile. What we need to remember is, in case of lower organisms or in case of lower plants, male gametes bilak kodai motile hoy. But we are talking about what? We are talking about angiosperms, and angiosperms are supposed to be highly evolved, right? Are highly evolved structure, like male gametes or female gametes thake, a gametes bilak non-motile. And since they are non-motile, সেই কাৰণে pollination লাগে. কোনোবাই এটা carry কৰিব লাগিব, এটা agency লাগে. Is the point understood now?
Yes, ma'am.
Okay, so answer কি হ'ব? Obviously it will be non-flagellate male gametes. Right? Okay. We'll move to question number 7. Okay. Polycyphinous condition of pollen grain is found in, right? So polycyphinous, pollen grain or related দুটা word আছে. এটা হল monocyphinous আৰু এটা হল polycyphinous. So we have a word called as siphon. What is a siphon? A tube-like structure is called as a siphon, right? So we are talking about the pollen tube that comes out of the pollen grain. So I think you remember we have drawn a diagram like this, no? So enzyme continuous, sorry, enzyme continuous আছে and enzyme works with certain germ pores, no? Etu ki boli koisilo? Germ pore. Aru etu ki asile enzyme. Our ভিতৰখন কি আছে enzyme, right? So germ pore function kiba, what is the most important contribution of the germ pore? It is that area through which the pollen tube comes out. Right? Hekiloke ami jano. So enzyme kon epidor পৰা যদি ulai hai ba epidor পৰা যদি ulai hai, that would behave as a pollen tube. Now the question is, eta pollen grain, or if one pollen grain has only one pollen tube, it is called as monocyphinous. One pollen tube. One pollen grain is always with one pollen tube, that is called as monocyphinous. One pollen grain, if it has more than one pollen tube, that is called as polycyphinous, right? So polycyphinous, I think we understand, more pollen tubes. in a pollen grain. Right? In majority of the cases, monocyclic condition is something that is very common. Samane eta pollen grain or etai pollen tube thakibo. But there are certain examples of plants where the polycyphinous condition is also seen, and the question has been asked about polycyphinous. Basic details of danibo nalage, keneke develop kore, kyo develop kore. These are something that is not required in the course. What you need to remember, just one line about polycyphinous is just the example, right? Basic example to night. So normal case of ki ho monocyphinous, polycyphinous condition ami code dekha pam. In case of cucurbitaceae or malvaceae. Right? Example, Malvaceae, one example of Malvaceae could be China rose, right? China rose is an example of Malvaceae. So question to the polycyphinous condition is seen in China rose. Similarly, if you're talking about cucurbitaceae, cucumber could be an example of cucurbitaceae. So just Malvaceae or cucurbitaceae, it is about monotrachylebo in case of polycyphinous condition. Next is that was question number 8. Here a question I read, 82% of angiosperms or angiosperm by angiosperm families have that type of ovule. Which ovule are most common? Anatropous, right? So up till here, what we have discussed is we have talked about certain types of questions that are supposed to be asked from this chapter. No, a part of the microsporogenesis, megasporogenesis. These are certain questions that you should remember. After this portion, the next unit that we need to discuss is pollination. Right? So pollination no hole, so fertilization no hole, so pollination is obviously an important aspect. So what we need to remember, or what we know about pollination, this is a very common topic that we have studied already. So what we know, pollination ki hoy? Transfer hoy of pollen grain. Transfer hoy from the anther to the polyzyte. Receptive stigma of the carpel. Right? Etu kami ki boli kom? Pollination boli kom. Next is, what is the need of pollination in case of higher plants, especially if we are talking about angiosperms? Okay, male gamete and female gamete. Okay. Flowering plants or থাকিব মেল গ্যামেট থাকিব আৰু ফিমেল গ্যামেট থাকিব ফিমেল গ্যামেট মেল গ্যামেট ক'ত থাকিব pollen grainত right আৰু ফিমেল গ্যামেট আমি ক'ত থাকিব we have already discussed in embryo sac no এতিয়া what another information that we know is these two gametes no they are non-motile so requires pollination agency for pollination or they undergo pollination no pollination হোৱা বা কোনোবা এটা বস্তু pollen বিলাক এটা জায়গাৰ পৰা আনটো জায়গালৈ carry কৰিবলে agency বা কিয় দৰকাৰ কাৰণ তাহাত non-motile হয় right next is type of pollination কিমান types আছে two types what are the two types self pollination and cross pollination right so pollination is two types self pollination and cross pollination Next is self-pollination. What is self-pollination? Self-pollination মানে কি? You have one flower and in that flower কি হয়, the male reproductive, female reproductive part is present, দুইটা বস্তু আছে and it gets fertilized. So same flower, same plant. এটুক আমি কি বুলি কওঁ? Self-pollination. So self-pollination is again of two types. Autogamy it is called as heterogamy. So what is the basic difference? Ek line will remember. Just will remember in one line, same plant, same flower, right? And heterogamy we have same plant but different flowers. এই দুটা লাইন আমি মনত ৰাখিব লাগিব. What is self-pollination? The pollination that occurs in the same plant or in the same flower, that is called as self-pollination. It has been divided into two parts, autogamy and heterogamy. So adult plant আছে, adult plant or you have a flower and in this flower both the male reproductive part and the female reproductive part is present. তাহাঁতৰ মাজত যদি pollination হয়, this is called as autogamy, right? Eto ki hobo? You have a plant, you have a flower here, and you have a flower here, right? Our pollination kopra hoise, yar pora yale pollination hoise. Tak ami ki boli kom? Heterogamy. So self-pollination, when we are talking about it, is the pollination that occurs in the same plant, right? And if you're talking about cross-pollination, then we are talking about pollination of two different flowers in two different plants, right? So if you have a plant A here, no, yate adult plant ase, yate flowers ase, and you have a plant B, yate flower ase, yate like yar pora yale jodi pollination hoy, tak ami ki boli kom? This would be an example of a cross-pollination, right? Dull plant or মাজত hoise. Jodi same plant or মাজত hoy, tak ami ki boli kom? Self-pollination. But while we are talking about self-pollination, we have to know about this term also, heterogamy. Same plant but two different plants. Jehetu same plant, he karone ami tak ki boli koisu self-pollination. Kintu it looks something like cross-pollination also, and that is the reason eta line monot rakhibo lage. Genetically, it is an example of self-pollination. A line to monot rakhiba. But ecologically or functionally, it is an example of cross-pollination, right? So this line is important. So two types, eto to problem type, same plant, same flower, pollination hoise, so that's called as autogamy. It is self-pollination. Heterogamy, eta problem ase ki, we have seen the pollination that occurs between two flowers of the same plant. The flower is not the same. So we have a statement that says, genetically, heterogamy is an example of self-pollination, but ecologically or functionally, it is an example of cross-pollination. Bujhi paiso? Are two terms clear? Now, right. So ultimately what we know is, if you are talking about a plant, so that ki hobo pare, cross pollination hobo pare aru self pollination hobo pare. So it is a plant that cross pollination hobo ne self pollination hobo, we have to talk about some consequences, right? So kisuman factors So jar karne plant self pollination hobo ba kisman factors jar karne cross pollination hobo. Adult plants or kisman factors kibo lagibo that will promote self pollination. And so we will be discussing certain factors that will help in undergoing self pollination. And what are the factors? The first one being bisexuality. What we understand by the word bisexual or bisexuality? Eta flower যদি দুইটা reproductive organs আছে, the male part and the female part is present in the same flower, that's called as bisexual, right? So if in the flower pollen grains আছে বা male part আছে আৰু plus stigma that egg বা that carpel আছে, so it becomes easy for pollination. So bisexual condition is a more important factor that always helps for self pollination. Adult plants or the bisexual flowers আছে, তেতিয়াহ'লে the first type of pollination that it will undergo or it will want to undergo is always self pollination. What is the reason behind? কারণ cross pollination হব লাগিলে তাহাতে agencyৰ কাৰণে wait কৰিব লাগিব. They have to wait for the agencies, so that will require certain amount of time. Instead of doing so, what would be easier? The easier will always be getting pollinated within themselves, and that is what is called as bisexuality. So self pollination hobo lagle, what is the factor that helps us? It's always the bisexual condition of a flower that helps in self pollination. The second is something called as homogamy. What is homogamy? Gamy means marriage, right? Homogamy, we are talking about a flower. That if this flower, suppose we have a flower like this, this is the thalamus, we have a flower like this, right? And this is the female reproductive part. Or etu hol male reproductive part, no? Etu hobo tomar anther, and this is the stigma, and this is this is the stamen, and this is the carpel. And what type of a flower it is? Bisexual flower. Okay. A bisexual flower yate ki ase? Pollen grains ase, yate ki ase? Egg ase. So if this comes here, তেতিয়াহ'লে কি হ'ব? Self pollination হৈ যাব. Now what is homogamy? If the pollen grain and the egg mature at the same time, means both of them are ready for fertilization at the same time, that is what is called as homogamy. Homogamy মানে কি? Both male and female reproductive parts mature at the same time. And the example could be mirabilis, right? So যদি homogamy হয় বা দুইটা যদি একেলগে mature কৰে, then it is easy that they will undergo self pollination. So this is the second factor that helps in undergoing self pollination. Next, we have something called as schistogamy. Schistogamy. Have you heard a word called that? Clistogamous flowers. শুনিছা নেকি? Clistogamous flowers মানে কি? The flowers that never open up, or closed flowers. Right? We have open flowers and we have closed flowers. What is the open flower? যদি তোমাৰ petals বিলাক হাতে মেলি দিয়ে, no? If suppose the petals or the petals get exposed, if the petals are open, then what happens is the reproductive parts get exposed. In that condition, we use the term open flower. But in certain cases, what happens? The flower remains closed. Means what? Petals ke khule na khai. Jodi khul na khai, pollination ki hobo? The pollination is something that will occur inside the flower. So in that case, in certain cases where the plants will have clistogamous flowers, tahator flowers will ketia open nohoy. Jodi open nohoy, obviously pollinators will nahibo. So what is the best method of pollination that they will undergo? It is always self pollination. So clistogamy is a factor that helps in self pollination. As far as examples of plants are concerned, here for good question ahead. Question to kya hai? What is clistogamy? Closed flower is something that you need to remember. Aru ki monot rakhibo lagibo the examples. Examples could be Viola, Commelina, oxalis, right? And Archis hypogaea also. Archis hypogaea কি বস্তু? Groundnut. So these are some common examples. তাৰপিছত comelinar ওপৰত বহুত question আছে. Another thing that you need to remember is এই তিনটা কেছত, in case of viola, commelina and oxalis, they will also have Casmogama flowers. What is Casmogama? Open flower. All the closed flowers থাকিব সেইটো নহয় যদি closed flowers আছে they will undergo self pollination যদি open flowers আছে they can undergo cross pollination. So this is the third factor. We are discussing certain factors that help in self pollination. Plantal self pollinate কিয় হ'ব এইবিলাক factors যদি থাকে তেতিয়াহলে self pollination হ'ব. And lastly we will be talking about the last factor, that is bud pollination. So what is bud pollination? In case of certain plants, what happens is pollination occurs at the bud condition itself, right? So কি হয় যে ফুলটো মেলিবলে নাপায় bud conditionতে pollination হয়. So pollination before opening of the flower occurs before opening of a flower, and this process is called as emphasis, right? So ultimately we have discussed certain factors that help in undergoing a self pollination in case of plant. So we are discussing about pollination. So the pollination, we understand that they are of two types. Adult plants, there are certain factors that favor either self pollination or factors that will favor cross pollination. Today we have discussed about self pollination and the factors that help in undergoing self pollination. In the next class, what is it talking about is cross pollination. Aru kunবিলাক factors karone cross pollination hoy. So I think this is all for today. Kiba doubt after the day, you can always ask me. After ni kiba doubt?
নাই ma'am.
নাই ন বুজি পাইছা বস্তুখিনি?
Ma'am.
Okay, so contributions খিনি ভালকে পঢ়িম, right? And in the next class, let's start with cross pollination. Thank you for today.
Thank you, ma'am.
ক্লাস শেষ নেকি ম্যাম?
ক্লাস শেষ, আপুনি হেৰি কৰিও এক্সিট কৰিব পাৰে। মইতো একো কৰিব নালাগে।